Good afternoon, everyone. Welcome to the H3D Foundation webinar series. Our today's speaker is Dr. Carolina Andrade. Dr. Andrade is an associate professor of medicinal chemistry at the Faculty of Pharmacy at Federal University of Goias, Brazil. Her research focuses on computer-assisted and artificial intelligence-oriented drug design, aiming at discovering new drug candidates for neglected and emerging diseases. Her research also focuses on the development and application of AI tools and QSR models for the prediction of toxicity properties of chemical compounds. Dr. Andrade has received several awards for her contributions. In 2016, she has been elected to the Young Academy of the Brazilian Academy of Sciences, and in 2021 to the Global Young Academy. Before we start the talk, the housekeeping rules that we'll have a Q&A session after the talk, so you can post your questions in the chat section or keep for the end of the talk. Today's topic is COVID AI, artificial intelligence-based platform for accelerating drug discovery for COVID-19. And now I would like to request the Dr. Andrade to share her work with us. Over to you, Dr. Andrade.
Okay, thank you so much, Ruti, for your kind introduction. So, first of all, I'd like to thank the H3D Foundation and the organizers of this webinar series. For me, it's a pleasure of being here today, talking a little bit of our work we have been developing in great part of this work I'm going to show you. It was developed in a multilateral project within H3D. So I'm very glad to be here today. So first of all, who and where are we? I usually like to start with this slide because my laboratory, LabMol, Laboratory for Molecular Modeling and Drug Design, is in Brazil, but is not in the most, populated or most known regions of Brazil. We are in the Middle East part of Brazil, near the capital, Brasilia. So we are in Goiás state. And our university, Federal University of Goiás, we have this new building of the Faculty of Pharmacy, where I am located in the 4th floor of this new building. We just moved in 2021, so this is our dry lab here, my laboratory of computers, and people, my students, my group returning from the COVID-19 lockdown. So we were very, very happy with this new space. And we also are part of the Excellence Center of Artificial Intelligence of my university. So the building of this SEIA is in another campus with a nice view also of the city. So I invite you all to visit us in Goiânia. Okay, and our research interests lies basically in two lines. So we apply and develop AI and computer-assisted drug design approaches for neglected and emerging viral diseases. And also we work developing in silico tools and platforms for predicting toxicity of chemicals. So today I'm going to focus more on the COVID AI project with viral emerging diseases. As most of you probably know, the drug discovery pipeline is a lengthy, costly, and uncertain process. So from each 10,000 compounds discovered in the beginning or in the early stages of this process, only 250 compounds go to the preclinical in vivo experiments. And here we have important steps with heat-to-lead optimization, ADME and toxicity and efficacy experiments in vivo models. And from those, only 10 go to the translational step with the clinical trials in humans. So less than 12% of all discovered compounds go to the clinical trials, and only 1 compound will be approved by the regulatory agencies as a new drug. So as you can see, this is a very long process within 10 to 15 years for one single drug to put in the market, and with a very costly around 2.6 to 13 billion US dollars for this whole process. So what do you need is to develop innovative approaches that could bring new drugs to the market faster and with a lower cost. And how can we do that? We are now using the artificial intelligence methodologies or approaches. AI is the simulation of human intelligence by machines, especially computers, on learning and problem solving. And the major field of artificial intelligence, inside it, we have some other approach within machine learning, deep learning, and also data science. So machine learning is the approach where computers can learn from data without being specifically programmed to it. And these approaches are now key components of everything we do nowadays. So where do we use AI? In almost every online shopping, digital speech assistant, and also with the self-driving car. So almost everything we use nowadays using computers, we use AI, and we even don't know that. Regarding the idea of using AI to study and to develop new drugs is based on the data that have been already created, generated. So we say that we are living in the big data era. It is estimated that there are 10 to 60 chemicals that were already synthesized, discovered, and cataloged in public databases. And from those, 10 to 33 compounds have properties like chemicals, so they are like drugs. So they are drug-like compounds, so they have physical, chemical properties like drugs that could be explored as new drugs. And also, we can use the public databases with compounds with at least one known bioactivity that has been already experimentally tested. So these databases such as PubChem, Campbell, have many compounds that we can use this information that have been already generated with the modeling approaches with AI to advance, to capture the knowledge that we need in this field. So the data, the experimental data, chemical and biological data have been already created, and it is in storage. But there is a huge gap in which we need to capture this knowledge and have a utility achieve. So we say that we are drowning in data, but starving for knowledge. And this gives us a tremendous opportunities for discovering new drugs, for learning with what have been already published, deposited, or tested, and trying to develop better drugs or better compounds with higher success rate, with optimized properties, and so on. So where AI is used, can be used in that whole process of drug discovery. In almost every step nowadays, AI is being applied from target discovery to post-approval approaches, even in the clinical trials. So AI is being used in all these steps of drug discovery, trying to optimize the process, to accelerate the process, and so on. And just to give you an idea how AI is changing the drug discovery process, this article was published back in 2018 in Nature, saying or showing how the big pharma companies are using AI, and they are performing collaborations or deal with startups with AI that are using these methodologies to accelerate or to speed up their discoveries. So during the COVID-19 run for drugs, antiviral drugs, there were this example of Benevolent AI, which is a UK startup company based on AI. And they discovered that baricitinib combined with remdesivir significantly reduced patient recovery time. And this went through phase 2 clinical trials. Also, this other company designed, not in the space of COVID-19, this was before the pandemics, but this company designed, synthesized, and validated a new candidate in 46 days. As we can see in this paper published in Nature Biotechnology, the company in silico medicine showed that from the target nomination, selection of the target, they use generative models and reinforcement learning models for generate new molecules for being synthesized based on the information, the previous information that have been already published and were in those public repositories. With these, they designed new compounds to be synthesized. These compounds were synthesized very quickly, and they performed the in vitro assays not only with the target, but also some metabolic stability and selectivity assays too. So this process took 46 days, in which a traditional hit lead generation takes around 3 years. So this is showing how AI can be defined, how can AI can change this idea of this length process to a quicker process. Within the computer-assisted drug design approaches, we can use many different computational approaches to accelerate or to help to aid the drug discovery process. Between these approaches, we can use ligand-based drug design approaches, LBDD, such as pharmacophore models, QSAR models, and the structure-based design approaches such as docking, molecular dynamic simulations, structure-based pharmacophores. So all these computational approaches are being fueled with AI algorithms to improve their predictions and to have better models for predicting the activity, not only the activity, but also the properties of compounds, pharmacokinetics and toxicity properties, to perform a multiparametric approach in this search for new drug candidates. One of these approaches is the KSAR, which stands for Quantitative Structure-Activity Relationships, in which we need, for developing this kind of computational model, we need chemical structures that have been, chemical compounds that have been already tested for some activity or some property that you are studying. So you use this database, this data set of compounds as training set, and we also remove some of those compounds as test set for a validation, for external validation process. With this, we try to generate models or equations correlating the variation of chemical structures of this data set with the variation of the biological properties. And how can we do that? We simply transform the chemical structure in a number, which is a descriptor. So the descriptor is a number, a util number that informs some structural pattern of these compounds. These molecular descriptors can be constitutional, topological, structural, mechanical, quanticals, fragments, and so on. There are thousands of available descriptors, and you can choose from 2D, 3D, 4D, 6D chemical descriptors. And with these, we construct the models using statistical, such as PLS and MLR statistical methods, or also nowadays we are building these models using machine learning and deep learning algorithms. And then the model will be validated with the external validation set according to a statistical process and the applicability domain. These QSAR models can be built as classification models, binary classification models for classifying active compounds from or differentiating active compounds from inactive compounds. And also you can build regression models, which will predict the biological activity of these compounds. And what is the utility, the main utility of these KSAR or AI models? If we can build predictive good models, statistically validated models, we can use them to virtual screening a chemical database of compounds that have never been tested against those target or that disease that you are studying, or with that pharmacokinetics or toxicity property that you are studying. So with those models, you can prioritize compounds should be experimentally validated. So from a huge database of compounds, from millions and millions of compounds, you can select a specific set, prioritized set for experimentally validating these compounds. And if these models are well defined and the applicability domain, and they are robust and predictive, you can have a high positive hit rate here in the experimental validation. So now I'm going to show you a little bit of our work in the COVID-19 approach. So together with Professor Kelly Shibali from H3D, myself and Professor Vadim from Russian Academy of Science, we jointly conceptualized an AI-driven approach for drug discovery for COVID-19. So we build on existing networks, and we additionally had some other project partners from Brazilian institutions and also from the United States, Dr. Sean Atkins from Collaborations Pharmaceuticals. And then we started this project within this BRICS call that was open in the beginning of the pandemics in 2020. So this project, this grant was approved. We called it COVID AI. And our main goal was to develop a platform with in silico artificial intelligence-based assays for 4 different targets, being the 2 proteases from the virus, the SARS-CoV-2, the spike protein, and also a human target, which is involved in the pyrimidine biosynthesis and is very important for the viral replication. So we develop all these models, these computational in silico models here in my laboratory at UFG. And with these validated models, we perform a different virtual screening approaches with these different filters with all the models and different libraries of synthetic compounds and also natural products, commercial databases, synthetic compounds from H3D, from Russian partner, from our partners from Brazil. And with these, we had some selected lists of compounds of virtual hits that we started testing in the target-based assays. After the results of the target-based assays, the experimental hits they are still being tested in the cell-based assays with the SARS-CoV-2 cytotoxicity assays, and our idea is also to perform the hit-to-lead optimization, the synthesis of new analogs optimized with potency and also ADME and toxicity properties. At the end, we hope to finish our project with more compounds being tested in vivo models and hopefully with new candidates, antiviral candidates for COVID-19. So when we started this project back in at the beginning of 2021, there were few compounds that were already tested for SARS-CoV-2 cytopathic effect in available databases. So we had at that time this screening, this PubChem screening with 365 compounds tested in this virocell cytopathic effect SARS-CoV-2. So we had this initial database for performing or for developing machine learning models. So we performed a rigorous data curation and modeling, and we had built classification models for classifying or differentiating active compounds from inactive compounds. So as we can see here, we tested different descriptors, molecular descriptors, 2D descriptors, with different machine learning algorithms to check which one performed better. So we had the random forest algorithm performing the best model, and this best classification model was used in the following steps that I'm going to show you in the next slides. So as I mentioned it, we divided this huge platform for drug discovery for COVID-19 in 3 or 4 sub-projects regarding the target. So we had the spike project, we had the proteases project, and we had the DHODH, the human enzyme project. So starting with the spike project, our idea was to use molecular, so structure-based approaches such as molecular docking with that machine learning models for SARS-CoV-2. And with these computational approaches, we started doing virtual screening based on FDA-approved drugs for repurposing. And then with that, we selected some compounds and tested those compounds in the enzyme, in the protein, and in the antiviral assays. So this was our first paper published within this work. We were able to find some beta blocker drugs such as carvedilol, carteolol, that showed interesting in binding constant for the spike protein experimental KD, and also carvedilol, atenolol, and bisoprolol were tested in the cell-based assays using Vero cells and using Calu3 cells, which are the pneumocyte human cells. And as we can see here, the comparison of carvedilol and remdesivir. So we have a very interesting starting point for drug repurposing in this scenario. Well, regarding the DHODH project, we have many different results. Some of them are not finished yet, and all of them we also didn't finish the paper. So I'm going to show you some of the results that I can show you now. This dehydrorotate deshydrogenase enzyme catalyzed the 4th step in the pyrimidine biosynthesis, which is necessary for the DNA biosynthesis. So it catalyzed the reduction of dehydroorotate in orotate. And we started, and this is a validated target for many human diseases such as cancer and arthritis, for example. And it's also very interesting target for viral diseases, as virus depends on the production of these pyrimidines for its replication. So we started with many data that was in the literature and in the databases for the human enzyme and human enzyme inhibitors. So our idea is to inhibit the human enzyme, which will show some antiviral effect on the replication of the virus inside the human cell. So we started with the search of the data of the structure of human DHODH and non-inhibitors, and we also performed the clusterization, the selection of these 3D protein structures from the PDB. We develop shape-based and QSAR based on machine learning and deep learning models for the non-human inhibitors of this enzyme. And with all these models developed, we started the virtual screening approaches using H3D database and also other commercial databases that we had access to them to prioritize compounds for the experimental validation. So we first started rebuilding or building new models for SARS-CoV-2 classification models using machine learning and deep learning, because new compounds, new data have been published and deposited in the public domain. So for this, we used a new data set of almost 10,000 compounds tested for SARS-CoV-2 in virus cells. And we tested in this case 2 different architectures for deep learning, such as feed-forward neural network and message passing neural network. And also we compared it with the random forest machine learning algorithm, which we previously found that performed better in the SARS-CoV-2 models. As we can see here, the feed-forward neural network with ACFP4 fingerprints performed better than the other 2 approaches. So we selected this model for the next step for the next virtual screenings. From the DHODH virtual screening using the H3D database of compounds, of synthetic compounds, so we have had access of these almost 7,000 compounds from H3D, and we started the virtual screening process by filtering step by step using the developed machine learning and computational models. So we use the information from the literature to develop and validate the computational and AI models. And with those models, we started looking at this database of compounds, which compounds could be prioritized for testing. So with those models, shape-based models, then machine learning models for DHODH, we also use the cheminformatics based inspection, and at the end we also screened these compounds in the machine learning model or deep learning model for SARS-CoV-2. So at this point we selected 36 compounds, and then 22 compounds were ready for being shipped to Brazil. So these 22 compounds have been experimentally tested in the enzyme with our collaborator, Professor Maria Cristina Nonato, and her team at University of São Paulo. And we were able to find in this dose response assay with the enzyme, this compound with a very promising initial IC50 of almost 3 micromolar. And as you can see, is a small compound. So our idea is to start performing the hit to lead optimization. We can grow this compound. We are also performing cell-based antiviral assay with this compound. Professor Maria Cristina succeeded to obtain a co-crystal of this compound with the enzyme. So they are working with this determination of this co-crystal with the human DHODH, and also new compounds that are derivatives of this compound that are already ready in H3D from others projects. They were selected and they are being shipped to Brazil for this next generation of trying to optimize this first hit discovered in this stage. As I told you, we have many other results for the DHODH, but I will just move more quickly here. Professor Flávio Emery from University of São Paulo with his team, they were working also in an alkyne series, and they were able to find this compound with activity in the nanomolar range of the enzyme. And this is the co-crystal structure, the crystallized structure of this compound within the active site of the enzyme. And also our colleagues in Fiocruz succeeded to experiment to validate this compound in CALO3 cells infected with SARS-CoV-2. They are repeating this experiment to calculate the EC50, but apparently it will be around 5 micromolar in the viral, inhibiting the viral replication. Within this DH, I'd like also to show you a little bit this new idea that we have been developing together with Sean Atkins from USA, of generating models, generative models that will generate new molecules for synthesis. So the idea is to use the classification models that we already, or regression models that we already built for human DH, and also the generative model with training all data available, so we can generate compounds and new compounds and evaluate those compounds first in the models and then synthesizing and testing those compounds. So Sean's team developed this generative model in their software called Megasim, and they optimizing the regression models for DHODH, IC50, also for solubility and cytotoxicity properties. And we think the priming molecule used as seed, that compound found from Flavio's group, PHM 307, that alkyne. So they were able to generate, to design many new compounds for being synthesized. So the idea, these are the predictions of the models, the activity IC50 and PIC50 here, cytotoxicity, solubility, and accessibility, synthetic accessibility. So with these generative models, the idea is now for Professor Flavio starting checking if it's possible to synthesize these compounds, and if yes, they will start doing that for trying to validate these generative models. So moving on for the proteases project. So we have also many results here, and some of them I cannot show you, but I will give you a brief overview of this project. The SARS-CoV-2 virus has two proteases. So one of them is called papain-like protease, PL-PRO, and the other one is the main protease, M-PRO. So the PL-PRO is has a palm, fingers, and thumb domain, and has a very flexible loop, which is called BL2 loop, very close to the catalytic site. In the catalytic site, there is the triad of the catalysis, and most of ligands or compounds that are found here, they are covalent inhibitors. And in our case, we were not looking for covalent inhibitors, we were looking for allosteric inhibitors in this BL2 flexible loop. So the first idea was to generate models, computational models, using the already available data for PL-PRO inhibitors. So we used the co-crystallized inhibitor in this BL2 loop, and with that, we developed docking models and validated those docking models with active compounds and inactives or decoys. And we also developed shape-based models using ROX, this software, which maps out, like a pharmacophore, a ligand-based approach for selecting new compounds based on this model. So we developed and validated these computational and AI models, and then we applied those models in the virtual screening approach for selecting or prioritizing compounds for testing, for experimentally validated. So we had different virtual screening based on the shape-based models and based on the docking models, using more than one PDB structure of the protein, as it is very flexible. So we use this approach called ensemble docking, which we can perform this analysis of different conformations of the protein from the crystal structures. And then with that, we were able, the compounds were selected and shipped to Brazil, compounds from H3D library. And with that, we were able to find in the first batch this starting compound with IC50 of 73 micromolar and Ki of 22 micromolar in the PL-Pro enzymatic assay. Within that, we found a derivative series of this naphtidine series, and we had found this heat compound here with IC50 of 15 micromolar. So further, around 30 compounds were supplied by H3D for these enzymatic evaluations. And we are now in the process of validating or doing other enzymatic and biophysical assays with the enzyme, with the PL-Pro, Ki determination, and we are also determining the antiviral, the cell-based assays, antiviral and cytotoxicity assays for the heat compounds on this series. The same idea was performed with the main protease of the SARS-CoV-2, but for MPRO, there are many more compounds and structures on the PDB available. So the main protease had, at the time we started this project, more than 300 PDB structures, 3D structures deposited on PDB. So how could we start SBDD, structure-based computational approach, with a PDB or a 3D structure of this protein? How could we select the best protein? So starting from that, we started with the target selection with the PCA analysis of PDB structures, which is very nice. So we can see the difference of the position of the amino acids and the catalytic sites, or the flexibility of these loops. So we performed this selection of the PDB, selected 5 structures, and then we performed the docking validation of our protocol using these 5 PDB structures with that idea of ensemble docking. So we used the 5 proteins in the same docking run. We succeeded to validate with active compounds and inactive compounds. And we also, in this case, there were many compounds that have been already tested for the MPRO enzymatic assay, mainly in a consortium called Moonshot Consortium. So we gathered all the compounds available, that more than 1,000 compounds tested against the MPRO. Nowadays, there are many more. Actually, we updated these models recently, but I don't have the results here. So we develop classification models and regression models for this data. So we have these QSAR models for MPRO, and also we validated or developed and validated these shape-based models, which is a ligand-based pharmacophore model, so we can map out the important characteristics of these known or experimental ligands. And we also performed this enrichment study with active compounds and inactive compounds. With those validated models, we performed the virtual screening, not only from H3D, using H3D database, but also from commercial databases. And in this case, we ended up with all these filters, with shape-based, ensemble docking, and machine learning and deep learning models. We ended up with around 40 compounds that were tested in the enzymatic assay. And in this case, we had these 3 initial hits in the enzyme. This is the control N3. So it's a very potent compound, but it is a covalent inhibitor. And our hits are not, they are allosteric inhibitors, which can be a good starting point for hit to lead optimization. So we are ready for this hit to lead optimization, and more compounds have been supplied from by H3D for this evaluation on protease. So a summary of our MPRO results. So we have many compounds with activity from H3D and also from commercial database. And we also lately performed a new collaboration with a group from Sweden in University of Gothenburg, in which they develop a MPRO yeast-based assay. So we could find that more hits from H3D are positive hits in this yeast-based assay, which can be very interesting for analyzing non-covalent inhibitors. So what we are doing now for this project, we are performing more biochemical and biophysical experiments with the protease, the main proteins. We also select some of these hits for the cell-based assays with SARS-CoV-2 and cytotoxicity, and we plan very soon to finish these results and wrap up in 1 or 2 papers. So with that, I think I'm done. I'd like to thank all the team, the COVID AI team within the H3D here. Most of you, I think, are listening to me today, so I'm very glad for being able to show a little bit of our more than 2 years effort of working together. And it's been such a pleasure of working with you guys and learning a lot with all of you. My team at LabMol, other groups in Brazil, such as Flavio and Christie's group in USP, Brasilia, Fiocruz doing the CALU3 and cytotoxicity assays. We also have our partner from Russia and other collaborators that ended up in the last year of this project, which is Professor Megan Shaw and Bianca from University of Western Cape, and also Professor Per from Gothenburg, and this group from Brazil, from also University of São Paulo, group from Professor Glaucius and Rafael, who are very experienced in the enzymatic and biochemical and biophysical assays with the protease. So they are also helping us with these new batches and new sets of compounds that are being tested. So this is our last meeting in Cape Town. It was a pleasure for visiting your beautiful country. And I'd like to thank all my team here from LabMol and do a little bit of advertisement and invite you all to submit your high quality research papers to our ACS Omega journal, which I am associate editor in the field of medicinal chemistry. So with that, it's just to say thanks to our funding agencies, our colleagues and collaborators. And again, thank you so much for H3D Foundation for the opportunity. And I'm very glad to answer or to discuss with you your questions. Thank you.